All right, good morning, everybody. Let's get busy, as they say. I think that's probably a cartoon character that said that. Okay. All right, we ended up with uh, checkpoints, right? So these are just to kind of recap from, let's get the chat up. Morning, everybody. These are super important because they are kind of like the quality control checks, essentially, of the cell cycle. And given that once a cell has divided, right, it can then divide again. Any issues or errors that are allowed to pass through that cell division essentially get incorporated into the daughter cells, right? So if you have some seriously messed up DNA and cell division still goes ahead, then each of those daughters will inherit most likely the same messed up version of that DNA, right? And so it's really important that we prevent that from happening, for one, and two, we make sure that cells don't divide when they're not supposed to, right? And so there are relatively few areas in an adult organism, like adult human, like you or I, that are dividing, right? I think probably limited to skin, hair, red blood cells, obviously, white blood cells like immune cells, intestinal cells, particularly in your stomach and your small intestine, right? They get a lot of wear and tear. Nails. I think that's about it. There really aren't very many other places that exhibit cell division, right? Maybe muscle or the like after injury or exercise, which is kind of the same thing, right? So most other parts of your body are not dividing, right? And they are not supposed to divide. They have no business doing that. And these checkpoints basically act as a series of clamps to prevent those cells from dividing again, right? Which is why obviously cancer is such a insidious and terrible disease, because it's essentially your own body doing something that it shouldn't. Okay, so make sure. Oh, one thing as well that I'm not sure if I stressed before, but it's worth pointing out again, given that there's exam opening today. Quite often I'll write stuff down in the chat in terms of definitions and information and problems and stuff like that. Those chat, sorry, puppy whining. Those chats are part of the material. Hey, come over here. Honestly, those are part of the material that are fair game for questions on the exam, right? Because they are part of the instruction that I'm giving you, an explanation of different things and bits and pieces. Okay, so don't forget those as you go through the material. You kind of put together your list of definitions and concepts and things like that. Okay. All right. So a little bit more on chromosome structure. This is actually pretty important. It's not always self-evident in many senses, right? So you're all familiar with the kind of the short and the long arm of chromosomes. We have P actually stands for petite. I don't know what Q stands for, perhaps because it's after P. But P is the small arm because it's petite. Petite is French for small. And we have telomeres at either end, right? These are the kind of buffers. Oh, all right. So I'm going to have to pick up puppy. Come here. Yeah. This is what trouble looks like. Yeah, it does. You go be quiet somewhere. All right. So hopefully she's going to settle down on the couch. Telomeres are essentially the buffers, and we'll get to this later on when we look at DNA replication. They deal with the end replication problem of linear chromosomes. So they're basically to protect your coding sequences against chromosome shortening, right? And then there's also the centromere. Now the centromere doesn't have any, well, I mean, I guess I gotta be careful when I talk about that, right? It's just DNA, right? So there's nothing especially different about the centromere, and it can occur any point, like along the chromosome. It could be right near one end or near the other end, or it could be right in the middle, or anywhere in between. Its main characteristic is that it is full of heterochromatin, right, which is inactive DNA, tightly coiled inactive DNA. And so there aren't really any coding regions, there aren't any genes, or very few at least, in that region of the chromosome. Oh, cool. Thanks, Piper. Or Rose, I'm sorry, I always forget what you prefer to go by. Yeah, so, but the key point is the centromere is the region at which the kinetic core assembles, right? So the kinetic core is essentially a collection of proteins, right, that recognize that heterochromatin at the center of the chromosome or somewhere in the middle of the chromosome. And they assemble around that, and that serves as a docking point for the spindle fibers, right? So essentially this part here, like if you look at the kind of the right 2/3 of this slide, this only occurs during cell division, right? So normally the rest of the time, actually this chromosome wouldn't even look like this. It would be long and extended out, look like a long piece of spaghetti, right? It's only during cell division that chromosomes condense this much. But anyway, after that chromosome's been replicated, you have these two sister chromatids, right? So this is still one chromosome, right? It's one kind of unit of information. Essentially, that's what a chromosome is. But these are two identical copies, right, as a result of DNA replication. These will then have the kinetic core start assembling to both sides, right? So we have to have spindle fibers attached to both sides of the chromosome to be able to pull apart those sister chromatids. And then these spindle fibers, which are made of microtubules, and that's essentially, it's actually really cool. They're very interesting fibers. And it's basically a series of repeating tubulin units. And these can assemble, they typically assemble at one end, the plus end. And so they can grow and contract, right? So they can extend, they get longer. It's literally like kind of, it's like making something longer in Lego, right? You just add bricks to the end of it, right? That's essentially how microtubules work. But you can also shorten them, right? So you can extend them to find those chromosomes, which is what happens during prophase. And then you can shorten them to pull those chromatids apart during anaphase. And also another thing to point out is that the kinetic core, right, where those spindle fibers attach, that's the site of the proteins that take part in the M phase checkpoint. Well, in part, kind of in between the sister chromatids and around the kinetic core. Okay, so these features they're important for cell division, but they're also important for some other aspects of kind of cell biology that we're going to talk about too. Okay, anybody got any questions so far about any of the cell cycle checkpoint stuff?
And you said that the kinetic cores were made from proteins, correct?
Correct. A whole bunch of them. I couldn't even tell you which ones in particular. There's more than I can remember.
Okay.
Yeah, there's a whole, they're both structural, as in they kind of clamp onto the DNA. They recognize that part of the chromosome as being the centromere, and they stick to it. And other things stick to them too, such as the spindle fibers. And they're also regulatory proteins. So those proteins are what determine whether or not you have equal tension on both sides of the chromosome, which is a signal for anaphase to proceed. But yeah, I've read about this like many times over the years, and like mostly it goes like in one end and out the other, and a little bit stays. So I have a basic understanding of how it works, but the details I don't know. Robert, you had a question?
Yeah, for the spindle assembly checkpoint on the cell cycle, what does that check for?
So that checks for, so the outcome, right? So it's kind of easier to start bigger. The outcome that it's looking for is every single chromosome, right, consistent of the two sister chromatids, is aligned on the metaphase plate, right? So essentially the attachment of the spindle fibers is random, right, from the centrosomes, and we'll get into some of those details in a bit. And so there's no guarantee that you'll have that attachment on both sides of the chromosome. So if you only have it on one side, that chromosome ends over on one side of the cell. So if you chop the cell in half, one cell gets two versions of that chromosome, the other one gets zero. And so what the spindle assembly checkpoint looks for, and that's part of that is what's called the anaphase promoting complex, which kind of does what you'd expect it says. What it's checking for is equal tension on both sides of the chromosome for every chromosome. And so just as if you've, I don't know, if you got like some small annoying younger brother or sister, and you and your brother or sister are kind of pulling on them from either side, you know, if you both pull equally, the little grot will end up in the middle, right? And that's kind of the principle that we're working on with chromosomes. If there's equal tension, then by definition, that chromosome is in the middle of the cell on the metaphase plate. And so what the spindle assembly complex, or the APC, looks for is, does that apply to every single chromosome? And it'll only allow anaphase to proceed if that is the case for every chromosome.
So once those microtubules have the proper or equal tension across all those chromosomes, then anaphase can proceed?
Correct.
Okay.
So anaphase is a really short phase of the M phase. Metaphase takes a long time because you're literally waiting for those chromosomes to kind of jiggle around and get properly attached and end up in the middle of the cell. And that takes time. But it's a pretty cool thing, really, because it works by mechanics, right? So it's not in a sense, it's not a molecular process, right? What they're looking for is equal tension. So there's some way of turning mechanical tension into some kind of biochemical signal, which tells the whole lot to proceed. Yeah, so it's pretty wild, right? And a lot of some of the genes that I work on in a dev bio class are actually part of that complex. So I've got like a passing understanding of how it works, but it's really quite astonishingly complex. Okay, so essentially in terms of cell division, or at least mitotic cell division, right? Again, got to be careful with language. Is interphase, right? Which we talked about kind of up here, yeah, which is all about preparing the cell for DNA and cytoplasmic division, right? So we have the preparation for DNA division in the GAP1 phase. So probably worth writing these down. Sorry, not DNA division, DNA synthesis. See, that's why I write stuff down. S is DNA synthesis, and GAP2 is preparation for DNA, I guess, DNA division, DNA and cytoplasmic division. Right, so all the things that are needed for M phase to occur, such as duplicating the centrioles and making more cell components, making stuff that's going to go into the cell wall, you know, all those different bits and pieces, those are all made in G2 or GAP2 phase, right? So each part of interphase has its purpose. And then really, the M phase is fairly quick by comparison, right? In terms of how much time each bit takes, this is a fairly simplified pie chart, but it's not too far off the ball, right? Kind of mouse cells take about 24 hours, I think, to divide off the top of my head. And of that, M phase takes maybe an hour to 2 hours, right? The rest of it is taken up by interphase. However, when we get into M phase for mitotic cell division, it's all about partitioning DNA, right? So we've duplicated DNA during S phase. Now we need to make sure that each daughter cell has one copy of each chromosome, right? So we start with our sister chromatids together. We need to separate those very accurately so that one goes into one daughter cell, the other one goes into the other daughter cell. And we end up with two daughter cells which are identical genetically to the parent. That's really, really important. Stroke the puppy a little bit. And these phases, so it's basically split into a series of phases, right? These phases all serve a different purpose, right? They're not, apart from anaphase, which is pretty distinct, right? They are kind of defined by what happens in them. So they're not like kind of like a timer on the toaster oven. They don't ping when one finishes. They're part of a continuum, but they all serve a different purpose, right? So really, the way I think of it, this might help you lot, is that you got to break stuff down at the beginning and get everything ready. You got to divide the DNA. You got to put everything back how it should be at the end, right? So prophase and prometaphase are the kind of breaking down and preparation phase of M phase. Telophase is really just tidying things up and getting stuff back to how they need to be for it to be an individual cell. So when you think of this as a series of events, kind of keep that in mind, because I've got to do that monkey face that someone told me to. I swear it's allergies or something, but you know, I take allergy pills up the wazoo, but it doesn't seem to help. And then the bit in the middle is really all about divvying up the DNA, and that's metaphase and anaphase, right? So you kind of got like a bookend. At the beginning is preparation, at the end it's tidying up, in the middle is where the DNA gets separated. And that's essentially what M phase of mitosis is. Meiosis is a bit more complicated. So good question, Adam. So telophase and cytokinesis kind of occur sort of simultaneously. That's why these things aren't like clockwork parts of a procession. They kind of blend into each other. So as telophase is going on, telophase is all about the DNA, cytokinesis is all about the cytoplasm, right? So cytokinesis is occurring kind of concurrently with telophase. And so cytokinesis is all about divvying up the cytoplasm and then basically kind of pinching off the cell membrane between the two daughters to create your two cells. It's kind of like if you put a rubber band around a balloon and it kind of shrinks together. And essentially, the final step would be if you could just go and you end up with two balloons. That's what cytokinesis is. The breaking down part really is actually prophase, right? That's where we break down the nuclear membrane and we condense the chromosomes and get everything ready. Telophase is kind of restoring everything back to how it needs to be in a functional cell, right? So it's kind of, yeah, M phase is a very weird kind of transitory period where stuff breaks down and weird stuff happens and then it just gets put back to how it should be, essentially. Okay, good question though. So these are the different phases. Obviously, I remember them as PMAT because my name's Matt. You could remember it however you like. Quite often it's taught as prophase, metaphase, anaphase, telophase. But there's also, you can kind of divvy up a little bit more finely with prometaphase in the middle, which is really kind of an intermediate between these two. Okay. Yeah, tying off, I sometimes call it like twins because you're going to end up with two identical offspring. The T bit is what I always struggle with in terms of thinking of a good acronym. Okay, let's make this smaller. There you go. So I can make this bigger. There we go. Okay, so essentially once the cell has gone through all of its preparation, right, it's got fully replicated DNA. The centrosomes, which consist of two centrioles, are duplicated because you're going to need to have one at either side of the cell for you to pull the DNA to those different sides, right? So these basically serve as anchors and the source of the spindle fibers, the microtubules, right? We have duplicated DNA in here. We still have an intact nuclear envelope or nuclear membrane, and so on, right? So without, you'd have to look real careful. You'd see, oh, we've got two sets of centrosomes. So this must be in the process of cell division, right? So this must be in G2. But if you otherwise looked at the cell, you wouldn't be able to tell that it's dividing, right? Because you wouldn't be able to see the extra DNA in the nucleus. It's just, you can't see it, right? It's like looking at a bowl of spaghetti from, the top of a building. You can't distinguish the individual strands. They all look the same. So even though the DNA is replicated, it has not yet condensed, right? And that's really important because if you can imagine trying to separate two portions of spaghetti equally, it's really hard. Counting out meatballs, on the other hand, is really easy, right? And so you want to get that DNA into a more manageable format. And that's really what prophase is all about. So in prophase, we have centrosomes. The centrosomes are starting to push themselves apart, right? And so they end up on opposite sides of the cell because they literally push each other as far apart as they can get with these spindle fibers, right? So they push themselves to the other side of the cell, but basically once they get there, they kind of anchor themselves to the inside of the cell membrane because they're going to be applying a lot of tension to those spindle fibers. So you don't want to pull them off of the cell membrane. They're going to be stuck there on either side. It's kind of like they stick out some microtubules out of the backside of them and attach themselves or glue themselves to the inside of the cell membrane. No, Adam, the nucleus actually disappears. That's the cool part. That's in prometaphase. So essentially, if you think about it, you've got some requirements for DNA division to happen. One, you need those centrosomes on either side. Okay, well, could I type these down? Another 3-point plan, right? I should be in like dieting or selling, I don't know, Peloton things on TV or something. Okay. We'll get to that question in a second, Adam. So first step, centrosomes need to move to opposite sides of the cell. 2, chromosomes need to condense. And then 3, how are those spindle fibers going to get to the chromosomes, to those kinetochores to attach if the nuclear membrane is still there, right? So the third requirement for DNA division to occur, this is all in prophase. Well, prophase, prometaphase. And good question though, Rose. 3, the nuclear membrane must disintegrate. All of this happens in prophase, which is kind of the condensation part, and prometaphase. So this is all of the preparation that's going to occur so that that DNA can be divided. And sorry, just getting back to Adams. Oh, so for the nuclear membrane, so essentially all of these processes are reversed in telophase. It's like a country and western song played backwards, as the joke goes, right? You get your dog back, you get your wife back, you get your trailer back, kind of thing. So in telophase, and that, sorry, hopefully no one is like a massive country fan, which is probably a good bet in Texas, I guess. But anyway, essentially in telophase, the chromosomes decondense, plasma membrane kind of reforms, and the spindle fibers disappear. So telophase is very much the opposite of pro and prometaphase. But as Rose said, this is all about preparation. Oh, and another. Oh, good question, Rose. So this is, like scientists, we like categorizing things. We like kind of drawing little lines and putting that into one box and that into another box. And that's really what this is. Prophase and prometaphase are purely distinguished by what happens in them, right? They are essentially a continuum in the process, right? There's no like stopping, pausing, and then going into prometaphase. It's all just one continual process. Even metaphase actually is kind of part of that. So in prometaphase, the nuclear membrane disappears, it's gone, right? So it fragments. That allows the spindle fibers to attach to the kinetochore. And also at this point, those centrosomes are on opposite sides of the cell. So if you think about this as a kind of a continuum, right? So we've got prophase, those centrosomes start moving, right, to opposite sides of the cell. Prometaphase, they are on opposite sides of the cell, right? And they start shooting out their spindle fibers. Got another definition that I need to put down in a second. Prophase, the chromosomes start condensing down. Prometaphase, they have finished their condensation maybe. So that's why many times, particularly in like GenBio-1, for example, there's not really any distinction between prophase and prometaphase. It's all just one preparatory phase. But as we kind of want to slice that pie a little bit more finely, you know, prometaphase now makes its appearance. And it's really just an intermediate between prophase and metaphase. Does that make sense? Cool. But really, it's like the key is just keeping in mind what needs to happen for DNA division to occur. Right, you have to have the chromosomes condensed because otherwise you're never going to fish them apart. It's just not going to happen, right? You need that space, essentially. You need to have your centrosomes on opposite sides of the cell because you're going to have to pull those sister chromatids apart to opposite sides of the cell. And there's no way in whatever you're going to attach those spindle fibers if there's a nuclear membrane in the way. So you've got to get rid of that. And that's really what prophase and prometaphase are all about. Oh, and another quick definition. Centrosomes are also referred to what are called mTOCs, which are microtubule, if I can spell that, organizing, so that definition is used interchangeably, essentially. And so really, you know, the end of prometaphase is really kind of blurs into metaphase, because now what needs to happen is you need to make sure each chromatid has a spindle fiber attached to it. So for example, you see this one here on the right-hand side, pardon me, that has a spindle fiber attached to both sides. Sometimes you can actually have two attached to both sides. But the key is equal tension, right? So sometimes you have two on one side and one on the other, so you'll have it pulling further to one side of the cell. Other times, as you can see for the other chromosomes in this cell, they only have one spindle fiber attached to each chromosome, right? And so time has to kind of progress to allow these randomly fired spindle fibers a chance to hit a kinetochore, essentially. And it's really like firing off harpoons blindly, right? You just go, fire another one, fire another one. Maybe one of them will hit. If it doesn't, you just kind of reel it back in, start again, right? So that goes over and over and over again. And that's really kind of the segue into metaphase, right? So by the end of metaphase, right, the beginning of metaphase is basically this. At the end of metaphase, every chromosome has a spindle fiber attached to each chromatid, right? So if you look really, really closely, see if I can make this a bit bigger. Right, you can see the kinetochore on each chromatid. Each one has a spindle fiber attached to it. And because there's one on each side, or two rarely on each side, this is essentially going to have a tug of war between the two centrosomes over each chromosome. Remember, each chromosome consists of two chromatids, which are their identical copies. And this is also actually a really neat way of recognizing mitosis and the different phases of meiosis, right? So when you're separating the sister chromatids, which occurs in mitosis and meiosis II, they always line up on the metaphase plate like so, end to end, like you got your fingertips touching your fingertips. If you're looking at meiosis I, they're going to be lined up like that in pairs instead. So keep that in mind too. Anyway, so after a while, all of these chromosomes eventually kind of jiggle their way into alignment on what is a kind of a present, a non-existent thing called a metaphase plate, right? So there's nothing in the cell yet that will define that region of the cell. It's literally the equal distance between the two centrosomes. But this is where every chromosome will end up. And metaphase will continue for as long as it takes for that to happen. Because anaphase will not be allowed to occur until you have every single chromosome on that metaphase plate, and every single spindle fiber that's attached to a chromosome is at equal tension. At least compared to the one on the other side of the chromosome. Does that make sense? Any questions, just fire away. Yeah, exactly, Rose. So there's nothing that defines that region of the cell as where those centrosomes will end up. It's not like the centrosomes are moving there because there's something there that they're attracted to. They simply end up there as a consequence of the equal tension between the spindle fibers on both sides. Oh, so Adam, actually, that's a good point. In both animal and plant cells, actually, the consequence of that metaphase plate being there is that other proteins do actually assemble there. And those are involved in cytokinesis, or building the plant, the cell wall in plants. So even though nothing marks that metaphase plate as something special before metaphase, somehow, I don't actually know how, the consequence of having that there is that there's now a ring of proteins kind of around the periphery of the cell, right? And those are what kind of pinch together in cytokinesis to form two animal cells, or serve as the kind of the building blocks for the plant cell wall to kind of meet together and divide the two cells that way. So this metaphase plate does have consequences. It just isn't a kind of physical thing beforehand. That's kind of why I want to keep emphasizing that role of tension from the spindle fibers in centering the chromosomes. The chromosomes aren't going there. They're getting pulled there, like in a tug of war, basically. Just as the spindle fibers don't attach specifically, they don't get fired and like make their way to a kinetochore. They just get shot off randomly, and eventually they find something. So anyway, once this has occurred, right, now we have equal tension on the spindle fibers for each chromosome. Now we have the spindle assembly checkpoint, or the anaphase promoting complex. And what that does is it checks for equal tension, basically on either side of every chromosome. When it finds it, basically what it does, it activates a protein called separase. Kind of self-evident in its name, right? And what separase does is they chews separase, yeah, that's about right. Chews up a protein called cohesin, which we'll come across a lot more in meiosis, that holds together the sister chromatids. Chromatides, you know what I mean. So those sister chromatids are held together physically by a protein called cohesin. And that cohesin is protected by another protein called Shigoshin. See, I told you it was complicated. Oh, what does Shigoshin stand for? That's Japanese for something. Bear. Yeah, I think some. I can't find it easily enough. I think it's Japanese for guardian. I think you're right, Rose. Oh, there you go. Guardian spirit. There you go. That's cool. Thank you, Dipper. Yeah, something based on those. Yeah, it's Japanese for guardian spirit. So, which is kind of cool. You know, scientists can have fun sometimes, I guess. But anyway, I'm actually sure of the details, but as part of the activation of the APC, separase is activated and Shigoshin is inactivated, right? And that allows the cohesin to be destroyed. And remember, right, we still have tension. Oh yeah, that's cool. Thank you, Rose. You've got a better memory than I do. Oh, cool. Oh, there you go. I mean, it's all based in language somewhere, right? Even if we don't always know exactly what that language or what those words mean. Anyway, so those chromatids are always under equal tension, right? So they're always trying to be pulled apart, but the cohesin prevents that from happening. And the cohesin is protected by Shigoshin. So once that protection goes away and separase is activated, separase kind of chews up the cohesin, and now there's nothing holding those chromatids together, but there's still the tension, and that tension basically just yanks them apart. And it looks very much like that. It's like you had a rope tied around your waist and then someone yanked you. You'd look like one of these things, like that. And because the spindle fibers are constantly being shortened, they're basically reeled in to one side of the cell and the other. Yoink, exactly. If chromosomes could have like an expression, that's pretty much what they would say when that happened. And this is really fast. Like, happens like that, right? Because all that the cell is waiting for is for that equal tension to be attained, and bang, they get pulled apart very, very quickly. And that also results partly from these so it's the APC. So this is where you start getting into my kind of fluffy, furry, blurry knowledge of APC and the spindle assembly complex. So activation of APC, which is the anaphase promoting complex, basically inactivates Shigoshin and activates separase. I don't actually know the details of all of that. There's a lot of players involved, a lot of phosphorylation of different targets and stuff like that. And whenever I read about it, I'm like, oh yeah, totally, I understand that. And then half an hour later, I'm like, don't get that at all. Right, so it's just quite complicated molecular biology, which I don't understand well enough to try and teach you. And it's not really super necessary at this level to understand that. Because you really just need to know what results in those chromosomes or chromatids, then chromosomes coming apart. Does that make sense? And actually, it's really interesting, right, that trisomy and monosomy, which we talked about a little earlier, is, and we'll perhaps cover this again when we talk about meiosis in a little bit, is the result of this M phase checkpoint failing. And so, if I remember rightly, human oocytes, so eggs in women, are, yeah, I'm pretty sure they're kept in metaphase. I think they're kept in metaphase 1. Yeah, pretty sure that's the case. I'd have to double check to be super confident, but I'm pretty sure all, for the ladies that are present, all of your oocytes are in metaphase 1. And those oocytes were created by you, obviously, when you're an embryo, when you're a fetus, actually, right? So when you're inside your mother. And they're kept in metaphase 1, pretty sure it's metaphase 1, for your entire reproductive life. And so even though this can go wrong in males as well, and so it's not necessarily a female only issue that creates like Down syndrome, for example, you can see how keeping those oocytes kind of paused in metaphase for potentially 40 or 50 years, as time goes on, the risk of one of those accidentally going through into anaphase increases over time. And so that's essentially the underlying basics of why you have things like Down syndrome, for example. Which is really interesting to think about. It's such a fascinating part of biology. And so this is going back to Adam's question earlier, right? So now that we've got our DNA in opposite sides of the cell, we have a ring where cleavage can occur, right? So that's what this cleavage furrow is. We now have a centrosome on each side of the cell. We don't need those spindle fibers anymore, right? So those spindle fibers disappear. The chromosomes start decondensing, right? So if you compare them here where they're real short and stubby, and look at them here, they're now starting to get longer, right? Because they're relaxing back into their normal state. And the nuclear envelope is reforming. And so this occurs kind of concurrently. It's hard to separate them clearly as this cleavage furrow forms, right? And so essentially the end result, let's see if I've got a good picture, right, is that you have these two daughter cells. So you have each daughter has one centrosome, it has a nucleus with a full complement of chromosomes, and that nucleus, that nuclear membrane has reformed, right? So this is the end of the process, right, which is happening during telophase and cytokinesis. And also the hope is that, you know, each cell will get, you know, roughly equal amount of mitochondria, for example. So mitochondria undergo cell division 2 during cell division, which is kind of weird. right? Because you need those in both cells, and all the other various organelles and the like will also be kind of divvied up too. So you can see how telophase is really the reverse of prophase and prometaphase, right? Prophase, the spindle fibers are forming, chromosomes are condensing, and they condense, where are we at? Here we go. They condense further, spindle fibers attach, nuclear membrane disappears in prometaphase, and then in telophase, we have the opposite occur. Chromosomes decondense, nuclear membrane forms, spindle fibers there disappear. And it's also really important to get mitosis, not just because it's interesting in and of itself, which it is, but also you've essentially learned half of meiosis now. Because mitosis and meiosis 2 are very similar. The only difference between mitosis and meiosis 2 is that mitosis cells are diploid, meiosis 2 the cells are haploid. Everything else is pretty much the same. So you get like a 2 for the price of 1 learning deal. See, even says it's imaginary. One thing that's also kind of an interesting quirk, right? So if you look at any images of early cell division in animals, at least, what you'll find is that the metaphase plate, the plane of cell division, rotates 90 degrees for every cell division. And so the reason for that is, let's see if we go back up here. Okay, so you have your, actually this isn't the best image, right? These centrosomes should be here. So the centrosomes start at one point and they push each other to opposite sides of the cell. Like so. And after cell division, you'll have your centrosome up here, right? Now, when that centrosome, if that cell undergoes a further round of cell division, which if this was the first division of a zygote, for example, it would. Now you can imagine this centrosome is going to be duplicated. Those centrosomes are going to be pushed apart to, it's not going to be top and bottom now, it's going to be left and right. And so because the division, the plane of division is halfway between the two centrosomes, now the plane is going to be vertical, right? So in this image, centrosomes end up here, the plane of division is horizontal. Now for each one of those centrosomes end up left and right rather than top and bottom. Plane of division is now vertical. And that happens every round of cell division. So if you look at a dividing early embryo, you'll see that it goes pop, 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 like that. It kind of rotates the plane of division every time. It's pretty funky. What else? So that's what it looks like in animals. It's fairly straightforward. Cleavage furrow. Plant cells obviously need to create a cell plate, which you can see here through deposition of vesicles on that what was the metaphase plate. So you get the new cell wall, and that's pretty much it. So this is all about growth, cell replacement. So essentially what generates new skin cells and hair cells and intestinal cells and things like that. And also if you reproduce asexually, which we don't, that's also how you do it. Yeah, exactly. Oh, no. So mitosis equals 1 round of cell division to generate 2 diploid identical daughters. Good question though. Meiosis equals 2 rounds of cell division to generate 4 haploid non-identical daughters. That's why we have meiosis 1, meiosis 2. And you always need to be very clear about which round of meiosis you're talking about. No worries, it's a good question to ask. Okay, any other questions? Questions, got a few minutes before we wrap up the first go around. Actually really glad that I changed the exam schedule to give us no extra class period. Otherwise we would have been like scrambling like loons trying to finish everything in time. So remember the key points: interphase, preparation for M phase. So think about what needs to happen for M phase. You know, you need 2 copies of DNA, you need 2 centrosomes, you need a whole bunch of other junk or stuff. Sorry, it's not junk, right? You got M phase, which is DNA division. You got cytokinesis, which kind of occurs towards the end of M phase, which is cytoplasmic division, right? M phase is all about preparation, right? So getting the DNA into a state where it can be pulled apart faithfully, making sure that the cell is ready to do that, right? That's the M phase checkpoint or the spindle assembly checkpoint. And then obviously pulling apart, which is arguably the least interesting part of M phase. And then putting stuff back together again, right? Which is really telophase, and obviously cytokinesis too. So just keep in mind like what has to happen, because one of the risks about a lot of this stuff, and meiosis is even worse, is it's just a lot of like numbers and words and stuff, right? So if you don't keep in mind what each bit does, like the purpose of each step, it can be overwhelming, right? It's just like a bleh. It's like me reading a ticker tape or something like that from a stock exchange. It's just not going to make any sense. So keep in mind the purpose of each step, like what has to happen, what's it looking for, what does it need? And it will make it a lot easier to understand. And then all these details, right? Like Shigoshin, separase, and cohesin, they then help you understand like the mechanism of how it works. It's not just an extra bunch of letters and words and stuff. All right, everybody, I'm gonna take a quick break. Make sure a little bit doesn't peel on the couch. And I shall see you back in 15 minutes at 12:15, okay? Cool beans. Robert, I'll have to tell you what that means in the UK at some point. Yeah. It's very rude.
Oh no, am I in trouble now?
No.
Okay.
We're in the US, right? That's
Yeah.
totally fine. I'll tell you about it later though.
Alrighty.
Okay.
All right.
See you all.
See you in 15. Yep.